For this exam question, students were asked to write an essay on the specificity of enzymes. So talking about the specificity of enzymes is simply what entails that enzymes are not disorganized entities. All right, enzymes are specific. So just when you use this, all right, an enzyme has an active site, and that active site has a special shape that only a substrate that is shaped like that can actually fit in. You get? But when you're asked to write an essay on something, it's usually crazy because you have to now say everything you know about that thing. Okay? It's not all about answering. You have to impress the examiner. So talking about the specificity of enzymes, we'll be having an introduction. We'll be talking about the types of specificity. They're talking about what? The mechanism of specificity. Then lastly, we'll be talking about the factors that actually influence specificity. Not lastly, okay, biological significance of what? Specificity of enzymes. You get? All right, so let's run through this quickly. For the definition or introduction, we're saying that enzymes are actually what? Biological catalysts that speed up chemical reactions in living organisms, right? So their incredible efficiency and specificity are fundamental to life because an enzyme will only interact with a substance or a substrate that was meant for it. So because they are specific, that's why we have a steady body. You don't have random reactions in the body. Do you understand? So enzyme specificity actually refers to the precise interaction between an enzyme and its substrate. I've shown you guys, okay? So this is an enzyme. Enzyme has a special what? Space for its substrate to come and fit in. So the substrate that can fit in and be acted upon that enzyme must actually have the shape that the enzyme wants. You get? So it's just like the enzyme is setting standards. You get? All right. So for you to actually be a friend of the enzyme, you will actually meet those standards. You get? All right. So the substrate is actually the molecule upon which the enzyme acts, right? So the specificity is what crucial for maintaining the complex biochemical processes within a cell. That's why I told you guys we don't have random stuffs occurring in the body. So what are the types of specificity? There could be absolute specificity. Now, what is an absolute specificity? Some enzymes like urease act only on one specific substrate. Some enzymes can act on other substrates, while some is only one. Once that substrate is not there, the enzyme is just there. Okay, so this urease only catalyzes the hydrolysis of urea. Okay, and this showcases that there's absolute specificity. Urease does not act on any other thing except urea. Okay, so this type of specificity ensures that the enzyme catalyzes the correct reaction without interfering with or without interference with other molecules, right? So if enzymes were not specific, as I'm here like this, I will not be stable because different random reactions will be occurring in my body. You get? There's group specificity. Okay, so certain enzymes such as what? Hexokinase exhibit group specificity, meaning they act on substrate within a specific functional group. So they are not acting on one thing, but once any substrate is a part of that group, they can act on it. Okay. Then hexokinase can phosphorylate what? Hexose sugars. That's basically sugars with 6 carbon atoms, but does not discriminate between what? Glucose, fructose, and galactose, because glucose, fructose, and galactose all have 6 carbon atoms. You get? So you should know that as I'm talking about the different types of specificity, I'm giving examples because those examples are what will fetch you marks, even extra marks. Okay. Then bond specificity. You said that some enzymes like what? Lipase, they target specific types of chemical bonds. Okay. So lipase catalyzes what? The hydrolysis of ester bonds in lipids. Okay. Breaking down fats into fatty acids and glycerol. Okay. So lipase, once something is a lipid, lipase will act on it to break it down, no matter what type of lipid you get. So that one is bond specific. Okay. Then stereochemical specificity. We said that enzymes like what? L-amino acid oxidase, they displace stereochemical word specificity, acting only on substrate in a particular spatial arrangement. Or when you say something is stereochemical, you see that word, it has a specific shape, it has a specific way in which it is arranged. So for this enzyme, once something is arranged like that, it acts on it. You get? So this is vital for processes that would require the correct isomer, right? Like protein synthesis, which uses what? These L-amino acids. You get? Then what is the mechanism of this specificity? Why are enzymes, or how are they able to what? Attain these what? Characteristics. So enzyme specificity what arises from what? The unique three-dimensional structure of the enzyme, okay? Particularly the active site. I've been telling you guys about the active site. Can you see how uniquely shaped the active site is? Anything that is not shaped like that cannot fit in. You get it? So the lock and key model, which was proposed by what ML Fisher, illustrate this concept, okay? So in this model, the enzyme active site is a perfect fit, okay, for the substrate, okay? Just like when you lock your cupboard with a padlock, somebody with another key cannot just come and open it. All right, with a good padlock. I'm saying a good padlock though. All right. So the enzyme side is a perfect fit, okay, much like a key that fits into a lock, okay. However, this model has been refined by the induced fit hypothesis, okay. So they actually would rearrange that stuff, okay, and they say that some substrate they can actually rearrange themselves to fit into an enzyme, okay. Just like the keys now that we have master keys, right? And this actually suggests that the active site undergoes a conformational change upon substrate binding, enhancing the enzyme's ability to catalyze the reaction, okay. So what are the factors affecting this specificity? So several factors contribute to what enzyme specificity. When they say factors affecting, it could be that due to some things, an enzyme that was not supposed to be acted upon by one substrate can be acted upon the substrate because of some factors. And sometimes an enzyme that was acted upon by a substrate will all of a sudden not be answerable to that substrate again because what? There were some factors that came in. So amino acid sequence. So the primary structure of an enzyme determines a three-dimensional shape, influencing the arrangement of the active site. Okay, so if the amino acid sequence change, the enzyme has changed. You get the environmental conditions, right? So environmental conditions like what temperature, pH, and the presence of cofactors or inhibitors can affect the enzyme's conformation and therefore its specificity. Okay, so for example, pepsin works best in what? Acidic environments of the stomach, while trypsin function in what? Alkaline conditions of the small intestine. So we are now saying that what? If the small intestine becomes acidic, trypsin cannot be able to function. If the stomach becomes what? Basic, pepsin will not be able to function. You get? Alright, so genetic regulation now. Alright, so genetic genes encodes enzymes, and mutations can alter their specificity. Evolution can shape the enzymes to be highly specific, all right, ensuring that biochemical pathways proceed efficiently and accurately. All right. So this image actually summarizes the types of specificity, which we spoke about some, right? So what is the significance of this word enzyme specificity to life when we say biologically? So it is actually vital for many reasons, like metabolic reactions, okay? So specificity ensures that the enzyme catalyzes only desired reactions so that you don't have a haphazard body, all right? And this is preventing unwanted side reactions that could disrupt cellular processes. The signal transduction. So enzymes like kinases and phosphatases play key roles in what? Cell signaling pathways, requiring high specificity to accurately what? Transmit signals, right? Then drug design. So understanding enzyme specificity is what crucial in designing drugs that target specific enzymes such as what inhibitors for viral proteases in what antiviral therapies. Okay. So conclusion now, enzyme specificity is what a cornerstone of biochemistry, enabling precise and efficient biological reactions. You could also add that what therefore preventing unwanted side reactions. All right, so that's it for what specificity of what? Enzymes.